Welcome to NeurON Air, the official podcast of Neurotech U of T, where we bring you the latest breakthroughs in neurotechnology straight from the experts. From groundbreaking researchers and cutting-edge engineers to pioneering clinicians and bold startup founders, we explore the innovations shaping the future of neurotech.
We are on a quest to spark curiosity, fuel conversations, and inspire the next generation of neuroprofessionals. Whether you're a student, a tech enthusiast, or just neurocurious, you're in the right place.
My name is Talan.
I am Arnav.
And we are your hosts at NeurON Air. I'm in my fourth year studying computer science and neuroscience at the University of Toronto and proudly serving as the co-president for Neurotech U of T.
And I'm a third year student studying cognitive science and business at the University of Toronto. We are super excited to introduce you all to some truly passionate and accomplished individuals in neurotechnology. So tune in and get ready for a deep dive into the world of neurotech.
In this episode, we're joined by Dr. Taufik Valiante. Dr. Valiante is an esteemed neurosurgeon, scientist, and leading expert in neuromodulation and epilepsy research. He is the director of the Surgical Epilepsy Program at the Crumble Brain Institute at the University Health Network's Toronto Western Hospital, and the co-founder and co-director of the Center for Advancing Neurotechnological Innovation to Application, otherwise known as CRANIA. Dr. Valiante is also a professor in the Temerty Faculty of Medicine, Department of Computer and Electrical Engineering, and the Institute of Biomedical Engineering at the University of Toronto.
Hello, Dr. Valiante. Welcome to NeurON Air. Thank you so much for joining us here.
My pleasure. Great to be here.
Yeah, let's get started.
All right, so we're just going to get started. For the listeners who are maybe not familiar with neuromodulation, could you explain what it is and its significance for modern medicine?
Sure. So neuromodulation, when I think of it as a doctor, I think of it as a way of changing either brain or spinal cord or nerve activity with some kind of device that's either implanted within the body or that lives outside of the body. So I can give one example of either. So I think one example that people are familiar with is transcranial magnetic stimulation, is where you have a sort of this donut kind of thing that you put over the brain, over the head, and it's outside of the body, and it generates an alternating magnetic field, which generates an electrical field in the brain, and then alters brain activity that way. Mm-hmm. And then the other kind of example, which is kind of closer, near and dear to my heart, of course, are invasive technologies. And so, for example, deep brain stimulation. So deep brain stimulation is where an electrode is implanted directly within the brain. And as you know, and as we know, the brain is an electrical organ. It's an electrical device. And so we use the currency of the brain, which is electricity. And we use electricity then to change the activity of the individual cells, the neurons, towards some kind of benefit for the patient. So for TMS, for example, it's a common indication is for depression, for example. Mm-hmm. And for deep brain stimulation, a common example is Parkinson's disease. However, there's many conditions which can be treated with neuromodulation. Of course, near and dear to my heart is epilepsy. So epilepsy is another condition of the brain that can be treated with neuromodulation, as well as things that we're pushing forward scientifically, which are neuropsychiatric conditions. So we think about things like maybe substance use disorder, or chronic pain, or even neurodegenerative disorders like Alzheimer's disease. So it's really a way of sort of leveraging how the brain works, which is it's an electrical organ, and then sort of modulating activity electrically to change activity towards some kind of benefit.
Mm-hmm. Yeah, thank you so much for that introduction. I do actually want to shift towards epilepsy since we know it's your expertise.
I'd love to.
As a neuroscience student myself, I do know a couple of things about it actually. Like it has different etiologies and different manifestations in different patients, as well as a variety of treatment options, like from pharmacological approaches to surgical interventions and even through ketogenic diets.
Yep.
How can neuromodulation help with alleviating the epileptic seizures?
Yeah, so great question. So I can tell you kind of the current state of the art, and then we can talk more about maybe what the future holds, I think, in the context of some of the work that we're doing and some of the advancements in hardware and machine learning, et cetera, et cetera. Yeah. So neuromodulation for surety has a role in epilepsy. There are a number of different targets within the brain. When we talk about targets, we mean where the electrodes actually sit to change brain activity. And so there's currently three sort of approaches to neuromodulating the brain. So one is neuromodulating brain activity. So one is actually not a brain operation. It actually involves implanting an electrode around the vagus nerve, which is a nerve in the neck. It sends its axons, its connections up to the brainstem, and then sort of those projections go up to the rest of the brain. And the way, for most of these devices, we're not really sure of the ultimate mechanism by which they actually function. However, we know from a lot of experimental work that stimulating the vagus nerve can reduce the seizure burden in patients. We don't think of it as a curative device or something we tell patients that their seizures are going to go away completely. However, it can in certain patients significantly reduce their burden of epilepsy. The other kind of bucket or other approach is to place electrodes directly where the seizures are occurring. And the one device that's involved with doing that, or modulating the brain in that context, is something called the NeuroPace. And so this is what's called responsive neurostimulation. And basically there is that once the patient, you've sort of identified where their seizures are coming from, then you place electrodes in those areas. And then the device senses when a seizure is going to occur and it stimulates the brain accordingly. Now that all sounds fine and dandy. However, the science and the research actually shows that the device itself actually does a relatively poor job at what it's supposed to be doing. In fact, it generates anywhere from 600 to 2,000 false positive stimulations per day. So its ability to detect seizures is not great. So it actually works more like a very non-specific device where it just kind of keeps, stimulating the brain. And probably through like plasticity mechanisms in the brain is the way it probably reduces the frequency of seizures. And then the last one, or general approach, is when you think of sort of traditional deep brain stimulation, and there's three different targets in the brain that we will use depending on the type of seizures patients have. So the three main targets are all within the thalamus, which you can think of it as sort of this kind of blob in the center of the brain, which is really acts like a sprinkler system. It connects really all of the brain to all of the brain, so really this real hub of activity. And so there's three nuclei. One is the anterior nucleus of the thalamus, one is the centromedian nucleus of the thalamus, and the last is the pulvinar. And these kind of like, if you think of it sort of anatomically, I've just kind of listed them from front to back. And so you can think of seizures or epilepsy that occurs, more anteriorly in the frontal lobes or temporal lobes might be better it may be better to treat with anterior nucleus stimulation, whereas maybe seizures coming from the back part of the brain may be better with pulvinar stimulation. That's currently the state. We know we can do better. We know that, for example, as I gave you that example of the responsive neurostimulator, is that it really does a very poor job of actually knowing when a seizure is going to start. And that's kind of really the holy grail of what we call closed loop devices, which is the loop that's being closed is that you have something that senses brain activity, it decodes it somehow, computationally, of course. It figures out, the brain's about to go into a seizure, and now I'm going to stimulate the brain. And that part about decoding and classifying, identifying when a seizure is going to happen is called prediction. So it's called seizure prediction. There's a lot of literature around it. So it's a very active area of research. It's hard with the brain. The brain's a complicated thing. But, I think all of us as, you guys as engineers, me as an engineer wannabe, but of course having a computational background, the promise is that there'll be some kind of magic in the math, some magic that we can do from a decoding point of view that we can actually very reliably detect when a seizure is going to happen. But that same problem, like if you think about this idea of decoding, like telling when or identifying when the brain's going to go into a seizure state, is very similar to all the other types of conditions I mentioned. So let's take, for example, using a similar device in depression, where the person, different than the seizure, they slip in and out of these depressive states, but a much slower time scale. Mm-hmm. But the problem is the same, which is record brain activity, identify some kind of biomarker of a state, and then stimulate at that state to try to push the brain out of that state. So epilepsy represents a really great example of the general problem, which is basically a control problem, which is observability, observe the brain, decode it, and try to push it towards some of that target activity with stimulation.
See, that's quite interesting. So you mentioned predicting the next seizure through like decoding and all the other aspects, and I'm curious, does machine learning and AI currently play a role?
Oh, of course. I think,
Yeah,
this is the buzzword and the catchphrase right
yeah.
now, I think, for this generation. Machine learning in of itself is kind of fancy statistics. It's basically, a sort of a non-hypothesis driven way of classifying some kind of activity. The thing which kind of moves it away from the traditional kind of statistics we tend to apply to these kinds of signals is that you do your best decoding in some kind of high dimensional space. So the more parameters you have, as for anything, whether you're trying to train a neural network to recognize cats or whatever, the more neurons you have in that network, and the more cat pictures you have, really help to sort of do the class, ultimately the learning of it. And so it's the same for us. The thing that we run into right now, which is really kind of something that we're very actively engaged in right now, is that currently the devices that exist are very simple. They actually are very sort of linear decoders in a way where they look at a single feature. So for example, they look at the power in one frequency band, and they say, if the power goes above this threshold, then something is going on. If it goes below, it's okay. But we know the brain doesn't work like that. And we know that the more features, the better. And this is really where the promise of, I think, machine learning comes in, is where we really know that more features are better, but we don't yet have the hardware that can actually look at things in this kind of high dimensional space and then say, yes, I think something is happening. So I think the computational side in epilepsy, for example, has moved very far, because offline, like not implanted in a device, you have no restrictions on your compute power. You have no restrictions on your energy consumption. You have no restrictions on heating. Like, it doesn't matter. You can just do whatever you want, on your data set. Whereas if you have an implantable device, you're really restricted. And that's actually been some of the work that I've been doing for actually quite a long time with a colleague of mine in electrical and computer engineering, Roman Genov, and many of the students that we've shared together, which is really making energy efficient hardware to do what we all tend to think of as machine learning, but really machine learning on chip.
Wow, that's quite interesting. Pivoting to your career as a neurosurgeon, you've obviously performed various surgeries, neuroimplant surgeries here at Toronto Western Hospital. Do you mind sharing a couple of successful patient stories, or some patient stories that are close to your heart?
Sure. Well, it's like every patient's close to my heart. Of course, I
Of course, yeah.
kind of joke, but in a way, it's advice. Every morning, the day I have surgery, we of course go see the patient and we talk to them. And Selin, you were there, so you saw that whole process. And for them, it's their one day. For me, it might be my third OR day of the week, but for them, it's their surgery. And so I think, the one thing that I always try to remind myself is that, that one surgery for them is super, super, important. And so, everybody gets that same consideration. But, if I had to play favorites, it's probably I'd play favorites with people who I stay in touch with, and who I've known for a very long time. So, for example, there was a young girl who now she lives out in BC, and she had a very bad frontal lobe epilepsy. And typical to epilepsy, she was in her very formative educational period where she was in university. And as you all know, and as you have all done, when you're cramming and sleep deprived for your exams, it's actually, those are triggers for seizures. And so when she would study hard and she would really, cram and, do all-nighters, her seizures would get worse and worse and worse, to the point that she actually couldn't finish her degree. So I remember her, as a young person at the time with her family, and you see a young person with the promise of their future, but they're trying, they're really trying to get through, but their condition doesn't. And so she actually had a very extensive workup, including an operation which we do from a diagnostic perspective, which is to implant electrodes within the brain. So we implanted various areas of her brain. And it turned out that her seizures were all coming from her frontal lobe. And then she ultimately went on to that second operation, which is like the therapeutic operation, and had a portion of her frontal lobe removed. Following that, she went on and completed 2 master's degrees, and now works. Has had many very interesting jobs, both in film and in history, and she also used to do grant reviews and grant writing and stuff like that. So, and I stay in touch with her. I think it's probably been 15 years, if not longer, that I've known her. And so it's kind of amazing to see somebody who's, gone through all that, who can then become seizure free, and then sort of pick up from where they left off and, kind of achieve the things that they want to achieve. And so, and there'd be many, many, stories like that, but hers is particularly sort of typical in the sense of somebody who's had really every kind of test done to get them to that ultimate point where we find that area of the brain which we can remove, and then we do remove it, and she has a really great outcome.
That's quite moving. Thank you for sharing that. And I'm gonna keep in touch with you in case I ever need a neurosurgery.
I hope you never need my services.
Wow. Yeah, thank you so much for sharing. I guess one thing is, like, when we're in the science side of things, you really cannot see like how it affects the real patients in real lives, and these are real people that we're talking about. So it's amazing to see like its effect in action. One thing that I was curious about is that, so we know that epilepsy, like, in oftentimes it's comorbid with like other diseases like depression too, right? So once you're trying to treat it, or like let's say you did like the frontal lobe removal surgery, right? How does that affect other functions?
So, I mean, that's a great question. Comorbidity is, in of itself, sort of already puts your brain in this kind of state where you automatically assume that the things that what they're struggling with are interrelated. We know that, let's take like mood, for example, depression. I could safely say that many of us, maybe you're too young yet, but life will throw enough curves at you at some point where you may experience depressive symptoms, or you may go into a period of depression. And so, our life experiences and what we're dealing with can affect us, from a mental health perspective, independent of seizures or epilepsy. However, we do know that about a third of patients who have epilepsy have a major affective disorder, which means that they are treated for it. And that can be either depression or anxiety, and they can also coexist. Having said that, you might say, well, are they completely separate then? Like, or do they actually coexist? Do the seizures drive depression? We don't have enough data to say that there's a direct link between epilepsy and seizures. But we do know that, of course, is when you have epilepsy, you have a lower quality of life. You can't drive. You're more likely to be underemployed or unemployed, right? You're probably not going to be as socially active or socially engaged for fear of stigmatization. And in fact, stigmatization is a major determinant of these individuals' quality of life. And so you can imagine all these kind of life events that conspire because of the condition that you have is to put you in a state of being, which nobody would want to be in that state. Like, you wouldn't want to be so restricted as a young person. And as I told you, as I mentioned to you about, the young patient that I talked about, despite her best effort to study and do work, the seizures got in the way. So it's like this external force, which is, forcing your life in a certain direction. But maybe there is. And so, there's some evidence where there may be an interaction, for example. So, for example, if I operate on a patient who has a pre-existing depression, and particularly if I operate on the right side of their brain and the right temporal lobe, it'll actually make their depression worse. And their depression will be worse for a transient period of time, and then they'll improve. Then it'll improve over time. So I think the message that we often give to patients is, and I think something I think we all can sort of take or decide what our mindsets are when we are faced with adversity is, do we just feel like passive players, as like, this is epilepsy, it's causing this to me, it's causing me depression? Or do we take an active role and say, I can work on these things independent of the condition that I have, and say I can implement, positive things towards changing that. So the word comorbid is a bit of I have a bit of a tension with it, right? Because in some ways it basically removes agency from the person, their sense of autonomy. You say, yeah, you're depressed. It's just part of epilepsy, right? So, yeah.
Well, thank you so much for that answer. So, shifting gears a little bit here,
Yeah.
we want to talk about neuroplasticity, right? So, it's brain's ability to reorganize and kind of like adapt to different input over time. With that in mind, what role does neuroplasticity play in the long-term effects of neuromodulation?
Yeah. So I think the role and the relationship is very intimate. I think what we really want to do when we're modulating the brain is not only modulating it immediately away from some kind of bad state, but hopefully modulating it in time as well, such that over time you might have to modulate it less, or there may be plastic changes that move it into a state which actually has either a less of an impact on the patient themselves from your clinical perspective, or you may have to modulate them less, so you have that burden of the stimulation is going to remove somehow. But really at the heart of it, when we think of neuromodulation, it really is about, leveraging plasticity mechanisms. And so I know most of the people who are listening, and probably yourselves, is when you think of plasticity, you think about synaptic plasticity. But there is another side of plasticity, in fact, something we study very actively in my lab, which we call intrinsic plasticity. And both of them, though, so synaptic plasticity is how the weights of the synapses change over time depending on the stimuli. And then the biophysical or intrinsic plasticity is how the biophysical properties of neurons change given their past activity. And so we really have this kind of presynaptic and postsynaptic side of plasticity. And really, I think neuromodulation likely affects both sides of this equation. The only hard part that we have is because right now the way we stimulate the brain, which is with electricity, it's relatively non-specific. And so we can't really make predictive models as to what exactly are we doing to that circuitry. But, very kind of broadly, I can sort of tell you my view on what neuromodulation does in the context of epilepsy, which is, so imagine you have a system that has plasticity, and typically the way plasticity works is that if a neuron thinks it's firing too much, it's going to make itself less excitable. And if it's not firing enough, it's going to make itself more excitable. So with that simple model in mind, you can imagine a scenario where if you have neuromodulation, like you have something stimulating the brain chronically, so you're driving up, in the short term, you're driving up excitability in a population of neurons. What are they going to do? They're going to slowly over time decrease their excitability, which means overall you're decreasing the excitability of the brain, which arguably then should help or prevent against seizures, because seizures is thought to be this hyperexcitable state of the brain. Now in Parkinson's disease, the circuitry becomes much more complex, for example. So can we really invoke those types of plasticity rules? We tend not to, in a way, partly because of what we see when we do stimulate the brain with deep brain stimulation, which is when the stimulator's on, their symptoms are good. And when the stimulator's off, their symptoms are bad. And so basically you have this immediate, it's got a very direct link between device on and off, which would argue that at least for the moment right now, our neuromodulation approaches are not leveraging the plasticity, which possibly we could in the future.
I see. So we have like a little bit of a coupling between the device state and the brain state, I would say, right?
100%.
Is that accurate? Yeah.
It's loose right now because, of course, the device is stimulating the brain and changing stuff, but the device doesn't really have conscious access to what it's doing to the brain.
Yeah. Thank you so much.
On the more sort of bright side, what are the biggest breakthroughs or advancements in your modulation that you have witnessed throughout in your career or even beyond your career?
Yeah. So I can tell you that the field itself is right now is a little bit sort of stable. I think it's wanting for newer technologies. I think there's a number of things that are moving the field of neuromodulation forward. I think one big one is imaging. So our ability to better image the brain, better able to image its connections through diffusion tensor imaging, and then merging these imaging technologies to targeting with when we target these electrodes, I think will bring important advancements in the clinical care of patients. So you might say, well, what do you mean by that? So, so let's take a hypothetical scenario. Let's say you're a brand new neurosurgeon. You just started your practice. You've only done a couple, 50 surgeries, for a specific condition you want to treat. However, you work at an institution where basically they've now have this huge repository of patient data, patient outcomes, imaging of where the electrodes were. And now you just put your patient's symptoms into a system, you put your patient's MRI into the system, and lo and behold, and I think this is not pie in the sky, the output of that would be the electrodes will give this patient the best outcome if they're placed exactly here. And I think that's something we're very excited about. I think mixing modalities together, mixing not only the device itself, but all the clinical variables, all the imaging data together, this kind of multimodal approach, I think is the promise of big data. And I think it's really the promise of that we're super excited about because we have yet to tap into those types of things. So I think that's one huge thing that we're all excited about. I think the other thing is, and just to be maybe a little bit egocentric, if you want to call it that, is kind of the work that we're doing, where really we think we've developed, intellectual property around really the next generation of chip technology, which really does look at the brain as a network, that does look at, hundreds of features at the same time. And I think, ultimately seeing the commercialization of a device like that, I think will bring a huge change to the field. Because then, in fact, for conditions, let's say depression, for example, where studies have shown that you can most accurately decode the depressed state of an individual by using 2 features in the right hemisphere and 3 features in the left hemisphere, and 2 of the 3 features go up and 3 of the 5 features go down. You can imagine now there's no device that does that. So I think that's another huge thing I think we're all excited about, that if those devices ultimately end up being realized, then I think that's a huge thing for us. And then I guess, the third big thing, which is the promise, which I don't think we've really realized, is true closed loop technologies. So the idea that you have really that device that's really able to very accurately decode brain state and then stimulate accordingly. But then you can imagine that that's only one dimension which you're recording from, which is an implanted device. Let's say now in the future, we have actigraphy, data coming to the device, and we have the patient and the time of day, because we know seizures occur at different times of days. So now you can just imagine as we start to, plop all this, data together. So I think that's super exciting in this field. I think that's really the aggregation of large scale data.
Yeah, that sounds like quite an utopian future, to be honest. Yeah, it's quite interesting. Yeah. Right. In terms of accessibility, what are the main barriers of making neuromodulation therapies more widely accessible to patients, and what solutions are currently underway to address these barriers?
Well, I mean, I can speak to epilepsy directly because it's something that I've been involved in a lot, specifically around access to care. And so for a patient ultimately to come to some kind of surgical intervention, they have to come down a very long sort of diagnostic process. So of course they'd have to be treated with medications. If they don't respond to medications and they're sent here, then we do a whole bunch of tests, whether it's an MRI or neuropsychology. They have a 2 to 4 week stay in the epilepsy monitoring unit. And then ultimately, if we have enough data, then we sort of say, yes, you're a potential surgical candidate. If we can figure out exactly from where the seizures are coming from in the brain, then we would offer them an operation to remove that part of the brain. We could either do that surgically, like what we call open surgery, or we have other technologies like laser interstitial thermal therapy, or what's called LIT, to actually put a laser fiber down to an area which we want to ablate or remove, and then we heat it with the laser to sort of remove it as another sort of surgical option. And when the surgical option's not there, like that surgical option is not there, like what we call resective surgery, which is resecting or removing a portion of the brain to help them with their seizures, then neuromodulation becomes an option. And so the rate limiting step in somebody getting to neuromodulation is really their ability to flow through this very complex system. And this complex system was something that I was involved in organizing in the province of Ontario. And very proudly in Ontario, we have the only regionalized approach to patients with epilepsy. which is really amazing for their care. However, just like everything in our life, we exist in finite systems, right? And so we don't have an infinite amount of funding or healthcare dollars to treat everybody who we want to treat, essentially. And so basically, patients get funneled down this thing, and one of the rate limiting steps is actually their referral to a surgical center like ours. So there's a number of process barriers, a number of sort of therapeutic and management barriers that patients have to overcome to ultimately get to that treatment. Now, once we're determined that there may be benefit from neuromodulation, we don't see a particular barrier in them accessing it because it's what we provide here at the Toronto Western Hospital. There may be some barriers in the context of the fact that we get a fixed amount of money from the government to do a certain number of cases. And so there is a bit of a constraint kind of on the volume or the number of cases that we can do. But fortunately, we don't often exceed those limits internally at the hospital, because in a way there's a bunch of filters that filter patients down to this sort of one therapeutic option.
I see.
Right, so we've discussed like a couple of methodologies in terms of neuromodulation, some of which are really invasive. I mean, most of them.
Yeah.
And in this sense, with this like growing interest and like research funding in neuromodulation-based research as well as therapies, there must be some important ethical concerns as well as the regulatory side of things, right? Could you maybe walk us through some of the key challenges or concerns in this sense?
Sure. So I can kind of maybe take you through different pathways. So let's pretend, we have a patient who comes through the epilepsy program. They end up being a good neuromodulation candidate, whatever it is, whether it's, vagus nerve stimulation or deep brain stimulation. Those devices are not experimental devices. They are, FDA Health Canada approved devices. So we don't consider these as sort of experimental things in these patients. And just like every operation we do, it ultimately comes down to the patient. And so, for example, we will sit with the patient, we'll explain to them the possible benefits of the device, the possible risks of the device, and ultimately it'll be their choice whether they want to do it or not. So when we propose a therapy in the context of neuromodulation, in the context of epilepsy, or let's say Parkinson's disease, these are on label, which means, the device is being used for what they were designed for. They're indicating that patient. There's an acceptable risk profile. The patient understands the risk and benefits of the procedure, and then they ultimately consent to the procedure. Now, there's another kind of pathway, which is really when we think of pushing the boundaries of neuromodulation. So what about a patient who has a disorder who in which neuromodulation wasn't considered, and we think that neuromodulation might help? And that's a different pathway. And so that pathway is really a research or experimental pathway. Let's take deep brain stimulation as an example. So, we have currently a trial now for a novel target in the brain to treat Alzheimer's disease. And we think that this approach is disease modifying, which means we actually think it'll, through neuroplasticity, change not only the structure but the function of the brain, and we think it can benefit patients. However, it's not commonly done as a standard of care for someone with Alzheimer's. We don't do that as a standard of care. So how do we go about that? So first and foremost, the trial has to be approved or has to pass sort of Health Canada approval, in the sense that the trial that we're doing is not an off-label use of the device. So for example, and I'll be maybe a bit, maybe jokey, but it's not like we're sticking the electrode in somebody's eyeball. We're sticking it in a part of the brain or parts of the brain that we've previously placed them before. So we're not using the device in a completely wholly solely new way. So in that way, the regulatory bodies are, okay, it's fine. You're using the device the way it's supposed to be used. However, if the device hasn't been used in that condition, then we have to submit to ethics here. And ethics will then review it. It helps, for example, in this case, we have peer review funding, which means it's gone through a set of experts who reviewed the trial who think that this is something important for patients with Alzheimer's disease. And so then ethics looks at the trial, specifically about like the process, et cetera, et cetera. We already have approval from Health Canada, so that's not an issue. And then ultimately, when it's acceptable to the ethics board, then we proceed down the trial. Again, now it always comes down to the patient, and the patient is the center of our attention. And so it'll be presented as a trial. And you can imagine for somebody with Alzheimer's disease, we don't want to create sort of vacuous hope for them. But we do want to say that, look, there's not really a lot of great stuff. They know this because there's really nothing to help with it right now. There's some promise of antibodies, but they have a very high risk profile, for example. So then it'd be like we presented to them as a study. We would not make promises about whether it works or not works, or it works or doesn't work. And then we would say, what do you think? Well, of course, answer the question. So there's a pretty strict way of, getting or doing the science that we want to do in the context of neuromodulation. Maybe the third thing, and I know that's a very long-winded answer, but the third thing might be is like, say you develop a brand new technology.
Yeah.
Say you've got a new device, what do you have to do? So it's a long road and it's a lot of money. So for example, to take a device from concept probably to an implanted device that you can implant in a human, FDA trial, approved trial, it's probably going to be anywhere from, $100 to $200 million. And many, many years, of course, of science and research, many years of device development, many years of animal work. So there's a lot that can sort of take, that you have to get through to get to like a brand new device for either an existing indication or a completely new indication.
I see. I guess on that note, as a follow-up, because I previously did work with like a new technological device, but this was for brain imaging. I was just wondering, what is the interplay between the industry, the commercial use devices that are kind of like being used in the research labs? Because I do know for a fact that they're super expensive, like even the development phase, and as well as like just getting that inside the lab is quite expensive as well, right?
Yeah. I mean, our experience with industry, we did work with Medtronic quite a bit. And their goals, of course, are to make devices to help people, but they also have other economic goals, right? They have to do things sustainably. They have to generate revenue so they can keep manufacturing devices, et cetera, et cetera. So oftentimes, industry is very risk averse. So if you wanted to use their device they're happy if you use their device for a different indication. So like, it is the way it is. We're going to plant it, but we're going to do it for a completely different new thing. They're like happy with that. What they're not happy with is like, hey, can we look under the hood of your device? Can we noodle with the software, the firmware, like whatever? And they're like, no. Because basically it's a whole FDA thing, whatever. And they're not really interested because that's super high risk for them. So it's understandable that we don't see a huge amount of innovation. We see a lot of innovation in the academic space, but we don't see that necessarily translated into a device, because just getting something into a human, like an implantable device, it's a class 4 device. So it's like the requirements are very stringent. All the testing you have to do is super stringent. The money, so the probability of starting with a brand new implantable device and getting to human is actually very low. Yeah. So I think that's why we see that, we're still using technologies that are kind of repackaged in a way for different indications, but the internal technologies don't really change a lot in the context of, existing commercial devices. And it's a real problem in this space, right? So,
I see.
All right, moving on to CRANIA. For our audience who does not know about CRANIA, could you tell us more about what is CRANIA and what was your vision as one of the co-founders?
Yeah. So CRANIA is, from a nuts and bolts point of view, it's a CFI grant, Canadian Foundation for Innovation grant, which are infrastructure grant, to bring infrastructure like robots and magnets and all MRI machines into the hospital here, to forward our sort of central vision, which is kind of translating neurotechnological advancements to therapy, essentially. And so that's kind of like the central, that's what it is. It was born out of the realization that here in Toronto, we have world-class engineering faculty. We have a long pedigree in neuromodulation. Some of the really important initial trials in neuromodulation were done here in Toronto. And we have really a world-class medical school. And so when you look at the field of neuromodulation, what you realize is that really it's a merger of these three disciplines. And although we had them here in Toronto, we really didn't think that they were sort of working synergistically together. And so CRANIA was this thing conceived by myself and Milos Popovic, who's now the chair of biomedical engineering and the director of the KITE at the Toronto Rehab Institute, which was to sort of bring together scientists in this kind of multidisciplinary space. And the catalyst for that was this kind of large CFI grant that we got to bring equipment towards that vision. The cool thing about what's happened with CRANIA, though, is that although all that equipment did come in, the one thing which was very exciting for us and something that we realized as possibly a need, and I think that in some ways you guys have articulated that very well and demonstrated that, given the interest in your club, for example, is that there's a real appetite for students in this space. And I think it's a very exciting space. I think one, it's brain. So for me, I have a personal, thing about that. It's cool. It's a hard problem. It's kind of on everybody's mind in a way because, we have our devices and we see stuff like AI and machine learning always remind us of the brain, right? We have advancements in, hardware, for good or for bad. We have things like Neuralink, right? Which, I won't speak to that directly right now, but so we have things in our spaces that kind of keep reminding us about the brain. And then it's a hard engineering problem. It's a material science problem. It's a computational problem. It's a medical problem. It's got like every facet, in this. So then we thought, well, in Toronto, there hasn't been a long sort of history in the context of neuromodulation as a scientific endeavor. Of course, we've used it as users of technology and we've done the clinical trials here, but we haven't really pushed that boundary. And who better than you guys? Because you guys are the future, right? You are the students who will, bring your excitement and your creativity and your abilities to this problem. So then we started to think about what we could do formally, educationally, to start to garner interest in this space. And so really we created the CRANIA Neuromodulation Institute, which is really the educational arm, if you want to call it, of CRANIA. And which I probably should have actually spelled out the acronym. So CRANIA is actually the Center for Advancing Neurotechnological Innovation to Application. So it took longer to create the acronym than write the grant, but anyway, so that's the acronym. But this educational arm was something we thought was really critical to drive this whole process forward. So through the CNMI, Cranial Neuromodulation Institute, we created a collaborative specialization. And so this is at the graduate level, where if you're doing a master's or PhD, you could actually get on your transcript ultimately that you did, sort of the subspecialization in neuromodulation. It would involve you taking an extra course, which we created, which is BME 1500, which is topics in neuromodulation. It's a cool course where we kind of survey sort of clinically applied neuromodulation in various different ways. We also survey briefly preclinical things like optogenetics and stuff like that. So things that we think might have ultimately clinical benefit in the future. So then we also then created, for me, which is, even, like the idea is to get you guys excited even earlier. You guys are already excited, I know, but even earlier. So we created EC441, which is electrical computer engineering, integrating and modulating the nervous system. I think I got that right. Where we introduced a little less clinical than BME 1500, more engineering style. I lecture there around oscillation, stuff like that, but it's a more engineering flavored. And there we have fourth year engineering students who have the option of taking it right when they're about to sort of, break off and think about what am I going to do, right? Am I going to go to industry? Am I going to go to academia? they want to build stuff, they want to do a PhD, they want to go into government. Like, so it's really this kind of thing which introduces you to this whole field from an engineering perspective. And now with the manager who I work with at the CRANIA Neuromodulation Institute, Sohail Asrar, we're now in the final steps of creating an undergraduate minor. There'll be a minor. So and I can't say this kind of more profoundly, I can't convey it in words, but I think that, for us, this field is super exciting. And I think what excites us the most is, of course, people like yourselves, right? Because we can then see the future in the work or the things that we've built here. We didn't build this stuff for ourselves. We built it, for people to use it and to sort of explore their abilities. And, our vision ultimately then is that we really start to elevate and get people excited in that space. So then whether you choose to do your graduate studies here or wherever, you're really set up for this very exciting field. We're seeing from an economic perspective that it's a growth area. So economically, we see it, growing, year by year, like 11% per year. This market analysis grows, right, around neuromodulation. So it's an important field, and I think we're just at the beginning of so many things. And we have many people here in Toronto who are doing neuromodulation. I mean, I talk about brain, but Paul Yu is studying and has a company around saphenous nerve stimulation for bladder conditions, for example. The work from Milos Popovic around functional electrical stimulation. Our work from invasive perspective. There's new technologies coming around, using non-invasive techniques like novel electromagnetic approaches. So there's a whole community in Toronto that is really doing this work. I mean, I even forgot to mention all the work that at Sunnybrook Hospital around focused ultrasound, as a novel neuromodulated technique. We have the Center for Addiction and Mental Health, which is a world leader in non-invasive neuromodulation, and we're seeing now the spin outs around computational aspects. So we really see Toronto as becoming this hub for neurotechnology, and I think CRANIA really that's its mandate. And I think the student experience and the student education is super, super important for us. Of course, we want to do great science as well, so we need to get grants and stuff like that. And so that's where we focus on seed funds for new partnerships between students, between different PIs where students can participate in. So it's become a really student-centric activity, which we're of course super, super excited about.
Yeah, it's clear you're super passionate about neurotechnology and sort of nurturing us to take over the reins, and you've got us hooked.
Oh. Awesome.
Yeah. But starting out any sort of initiative like CRANIA would have its fair course of challenges, and I'm curious, what were those challenges starting out?
Yeah. Okay, so I think I'll just mention one, which is maybe my personal one, which is, it's one thing to dream in a grant, and it's one thing to operationalize it. And so, getting, the rubber hitting the road, so I give one example, which is, we had this idea that there was a Canadian company that makes a novel MRI machine. And we thought that'd be super cool for neuromodulation because we could image the electrodes better in the brain. Mm-hmm. And, you can dream about the science and you can write it in the thing, but oh my God, I mean, the amount of effort it took to ultimately bring it to the physical space where it sat required just, a gazillion different departments, a gazillion different, leaders in different layers of the institution. It required a real commitment of the institution, which they had made. But it's one thing to commit and then actually see it realized, to the grant. And so, we're very lucky to have, great support from our CEO, Kevin Smith, our EVP of Research, Brad Waters. So, having people who are really, married in a way to the vision, was super helpful because it's not easy bringing a device into a clinical space for research, and then of course there's all sorts of other technical challenges, et cetera, et cetera. So that for me is like a great reminder is that, writing one thing in a grant is very different than actually ultimately operationalizing it. But I think the great thing you learn from this is that, that when you're able to bring people around a common vision, you can realize them. And I think that's the one thing, beauty for us in the context of CRANIA was that I think that vision was, something that I think a lot of people want to see realized. Um, there's going to be all sorts of things. Anytime you set your mind on, that kind of blue sky kind of thing or something really, there's always going to be those challenges. But I think the key thing is kind of knowing your why in the sense of like, what are the emotional things that keep you driven? And so for me, since I was young, this has been this deep desire to understand the brain. And so for me, that excitement can't be dampened by any kind of hurdle or administrative thing. It's just, this thing, it's just like when you're eating your favorite food, nobody's going to stop you from that. So it's kind of like the same kind of thing.
Well, yeah, thank you so much for sharing that. I do see like there are a lot of stakeholders, like you got to get a lot of things right, like the time, the technology, the correct people, the correct place to drive the innovation. So that was great to hear. I was just wondering, so you're a physician scientist, right?
Yeah.
How do you leverage the interplay between your clinical practice and groundbreaking research as well as innovation?
Yeah. So it's a great question, and it's very deliberate. The way I chose to organize my lab and my work, and it's really because of the fact that epilepsy as a condition, a neurological condition, offers us unprecedented access to the human brain. And so neurosurgery, my choice to do neurosurgery was really around that because when I was doing my PhD, I was I realized that if I were to ever sort of make something or develop something, I would want to be the one doing it. And so neurosurgery became a very natural, next step for me. And the surgery of epilepsy, as we briefly touched upon, is actually quite invasive. So, for example, if we're deciding if we found an area of the brain from which we can remove to help somebody with their seizures, we actually open their head and I remove that portion of the brain, which then actually allows me to study that portion of the brain. We bring it to the lab and we study very fundamental aspects of human neurons. Likewise, when we're working somebody up for epilepsy, where we have to implant electrodes within their brain to identify where their seizures are coming from, you can imagine that provides us unprecedented access. No other condition of the brain allows us to implant many hundreds of electrodes to understand not only where the seizures are coming from, but of course with the patient's consent and participation, is to understand how the human brain works. So a big part of my research, in fact, leverages our access to the human brain. We actually do a type of recording which there's no other place in Canada that does it, and a few places in the world that do it, which is we implant special micro wires in every part of every electrode that we are implanting for the diagnosis and management of the patient's epilepsy, which allows us to record from single cells of the human brain. And so now the patients are in the epilepsy monitoring unit. They'll often be there on average, 14 days or so, which then means we can not only monitor them from a clinical point of view, but of course with their consent and participation, they can choose to help us understand very fundamental aspects of the human brain, particularly things like that are related to memory, for example, because the one thing we can test very uniquely in the human, we can't test in any other organism on the planet, is what we call declarative memory, which is your ability to talk about your memory. And we can't do that in any other organism. We can't do that in a monkey or a rodent or anything like that. So what we choose to do is science that is very opportunistic in the sense is that we have this very unique opportunity to study the human brain. We actually then study things that are uniquely human. And we have that ability to study it from small circuits because we have the clinical electrodes all the way down to single cells. So the design of my research, in fact, has been, basically is the net result of a very planned approach and opportunity, which is the management of those with epilepsy.
Oh, that's really insightful to hear, especially like I think in a way you're kind of like having this clinical aspect to it is making even the research side multimodal. Like you get the clinical side of things, like
Yes.
electrophysiological recordings, but you also have the patient-centric view as well.
100%. And actually, you bring up a great point, which is, something as a clinician that, there's unfortunately times when I'll come back to a patient who we've done every test in and say, I'm really sorry, but there's nothing right now that we can do for you. And then even if we do something, so for example, we do some form of neuromodulation and has maybe some benefit, but not a great benefit for them, it'd be like, well, what's next? And, that's something too, which I feel very privileged because, they are also, they're not only excited, but, I think it's a human, very human thing, which is hope that we can concretely say, we are doing science, we are doing things that will help us, directly advance our understanding of the brain, or, building technologies that may ultimately benefit you in the future, which although immediately at that moment in time doesn't benefit them, at least creates that hope. And I think that's something as a clinician, I love, and I feel very privileged, to be able to do, in the lab and otherwise, is to sort of be able to convey that to them. And maybe on that note, I'll just make one plug, which is that on March 26th is going to be Purple Day, which is basically a global initiative to raise awareness for those with epilepsy. And in the atrium of this hospital, it will be all purple, and you'll be able to sort of experience all the research that we do at the hospital, and learn more about epilepsy. So I would say that I would encourage, you and your team and, anybody who listens to this, to come and join us on that day in the atrium of the Toronto Western Hospital.
Wow, that's great to hear. And I'm pretty sure there are a lot of interested people to come join and see the research firsthand. I guess as we wrap up this insightful conversation, I do have one last question. It's kind of more tailored towards the undergraduate listeners to this podcast. What advice would you give to young scientists and engineers who are eager to dive into the neuromodulation space?
Yeah, so that's a great question. And of course, rightly or wrongly, I think, I end up giving a lot of advice to students at every level. My advice is a bit generic, but I think for me it's been very important because as we've sort of talked about many things in this interview, and not the least of which is that, I'd like to paint a fully rosy picture and everything sparkles and rainbows, but it's not. And, and you struggle, there's going to be, points where you're working hard and you're just not getting to where you want to go. And then you have to pivot, et cetera, et cetera. So I would say for me personally, and I alluded to this, is really sort of know why you want to do this. So what is a question that burns inside of you? And I think that why is really an emotional thing, and it might come from something you've become passionate about in regards to maybe a family member has something, or something you heard of, or something about something really catches something, in you that makes you say, I really want to do this because of this. And that's a bit different than, kind of the reason why you eat chocolate ice cream. Maybe that's not your favorite flavor, but it is mine. Is, chocolate ice cream is kind of like, it's immediate. It's like, it tastes good. Now, of course, if you eat a whole liter of it, it's not going to taste very good after the whole liter. But that kind of pleasure or happiness is transient. The why question is something that keeps that happiness or that drive for long periods of time. So I would say that, I love, of course, the field of neuromodulation. I think it's a very exciting thing. And of course, I could, talk ad infinitum about it. But I think for the person, for that individual, for that student, is really find what, is your why. And then, and for me, when I was young, it was really to understand the brain as sort of nondescript and non-specific as it sounds. It's not like I had a specific mechanism in mind. It's just that since I was a teenager, so the brain fascinated me. And so, and then what follows from that is your how and what, because how you choose to do it could be different. You might choose to do it as an engineer. I chose to do it initially, my background was physics, which I thought, physics, the language of the brain. So I'm going to do physics, and then as a neurosurgeon, right? And then what you do, then you choose do I like to study things with MRI, or do I like things at very fast time scales like electrophysiology? So then you pick the thing that really helps you with your why, that really feeds into your strengths. And I think neuromodulation has literally something for everybody in a way, whether it's, material science, the machine learning or computational side of things, hardware aspects of the clinical promises, of helping. So I think there's so many things that could be, and I think it's an active process. It's a journey where you kind of, explore the space. It's like an optimization problem. It's like you explore around and you might fall in a low valley, and you have to generate a bit of noise and it pushes you out of that valley and you go into another thing. And so there's a very active exploration. So that's why too I'm super excited about potential partnerships with you guys and CRANIA, because I think we can put a little bit of a noise in the system. There's a lot of stuff happening, and that hopefully that why that once you get it is really sustaining. And the realization too is that getting to the why, it'll be many different kind of paths. So don't get fixated on the path. Really maybe really have that that sort of beacon for yourself, and things will change along the way. And so be adaptable as well. It's super important that what you might be doing at this moment in time, you may not see directly related to that long-term why, but it helps you feel like it's going to build something for you at that time. So as much as I like to sort of tout the whole neuromodulation thing, I think what I'd love to see our students who come with that thing is, I want to study this, I want to do this, like show me how I can do it. And I think that's something that we get excited about as well. So,
yeah. I really kind of relate to the point around that disruptive chaos, like the dynamic exploration process that kind of finally like leads you to the correct way in your life. But in the end, I would say like your internal drive is the thing that drives the innovation for all, I guess.
Exactly.
Thank you so much for this conversation.
My pleasure.
Yeah, thank you so much. It was really great to talk to you.
My pleasure. Thanks, guys.
That's a wrap for this episode of NeurON Air. We hope that you enjoyed this conversation and left with some electrifying insights. If you liked what you heard, subscribe to stay updated on future episodes, and don't forget to follow Neurotech U of T on social media for more cutting-edge content. If you have questions or topics you'd love us to explore, feel free to reach out. We'd love to hear from you. Until next time, stay neurocurious and keep pushing the boundaries of neurotech with us. This is NeurON Air, signing off.